Hello and welcome everyone. In this podcast, we're going to talk about modifying chromatin structure. In the previous couple of podcasts, we talked about how the chromatin was highly organized. And one of the structures of organization that we talked about in great detail was that of the nucleosome, the fundamental unit of the chromatin. And if we can just be generic here about genes, we know genes take up more than just that much space on a loop, but we're just going to draw it that way right now. The way I have drawn this right now, enzymes that are needed to come in here to turn this gene on, transcription factors, RNA polymerase, would have ready access to all three of these genes. However, in the process of compressing the DNA and in regulating expression, these octamers of histones bind around it to create the nucleosome, as we talked about last time. We'll put one over around this gene too. So as it's written now, only this gene is accessible. So the fundamental problem that we must address now is, how do we get to these buried genes? How is that regulated? Do we always want to get to those genes? Maybe we want to keep some of them buried. We'll talk about issues of it being tissue specific, temporal specific. Maybe at certain times of development, these genes will need to be on, and other times they need to be off. And so these nucleosomes need to be two things, which will almost seem a little contrary at first. These nucleosomes must be heritable. For instance, if this gene is needed during embryonic development, but not as an adult, we want that gene to be permanently turned off in all the cells of an adult. So every time an adult cell divides, this nucleosome structure around that gene, as well as some other structures in this area, need to be maintained. So it has to be heritable. It also needs to be reversible. We may have genes that need to be turned on and off at various times of development or at various times of life based upon certain situations the cell might be in. Maybe it's some kind of stress situation, we need to turn the gene on, or maybe during different times of the cell cycle, genes need to be turned on or off. So these nucleosomes need to be reversible as well. So we're going to begin talking about how we're going to have certain proteins gain access to this DNA that is protected against enzymes. How are we going to do that? And I'll give you a warning. At the end of these podcasts talking about this topic, we'll have to close with saying we don't understand exactly what's happening. Remember, this is still a relatively new field, particularly understanding the molecular characteristics of it. And so we've got a lot of really cool experiments that have been done and a lot of evidence pointing in certain directions. But it's still kind of a black box. We're still working on it. It's a pretty exciting time to be talking about this because of its early stages of development. Okay, I want to begin by talking about chromatin remodeling factors. Or we could say complexes. When we talk about factors or complexes, it's important to remember that it's not one protein. It's a large structure, so it is a large structure, protein structure, made of several proteins. For instance, when we talk about transcription factors, we're not talking about a single protein. We're talking about a lot of proteins around each other that provide a similar function. Each one of those proteins in that factor or complex has a specific role. And in some cases, they can be interchangeable. Sometimes you can move a subunit out of the factor, and now it has a slightly different function. Maybe one of these proteins in this factor provides tissue specificity. So keep that in mind, that we're not talking about a single protein. We're talking about many proteins coming together doing a common job. Okay, let's begin our discussion of these chromatin remodeling factors. I want to begin by talking about this large family of ATPases. These are going to be involved in ATP hydrolysis. So they're going to provide energy to that complex to do the specific job of remodeling the chromatin. There are many members in this family. I'm going to list 3 of them. The first is the imitation switch ATPases, which are abbreviated as ISWI. The second one is the chromodomain and helicase-like domain ATPases, and this is abbreviated as CHD for the chromodomain helicase domain ATPase. And this last one is the one we're going to spend all of our time talking about, or most of our time talking about, and that is the SWI SNF ATPases. Okay, they're all ATPases that are involved in chromatin remodeling. But as I said, we're going to focus our time on this one. So let's dig into the SWI SNF ATPase. And I'm going to focus, for the most part, with the one in yeast, because we know a lot more about that one. But there are homologs of this complex also in Drosophila and mammals. In yeast, we call this complex, after its original name here, we call it the SWI-SNF complex, SWI SNF. So it's after that one. In Drosophila, we call it BAP for BAP, and then in mammals, it's called BAF. Each of these are multi-protein factors, all with a central protein in the middle that is the ATPase. So in yeast, it is SWI2, SNF2. In Drosophila, does the same function, but it's called Brahma, BRAHMA. In humans, it's called HBRM for human Brahma. These all have the ATPase function and all provide the energy necessary for chromatin remodeling. And what I'd like to do now is focus our attention on the yeast version of this family. We know a little bit more about it, and your book also focuses on it a little bit more too. So let's zero in on this complex here. And I'm going to draw it over here real quick. SWI2 SNF2 complex here. And again, remember, this provides the ATPase function. Now this complex here in yeast will contain about 12 subunits, and some of them can be interchangeable. And I want to talk about some of them. I'm going to talk about this complex here called SNF5. In yeast, you know you're talking about a yeast gene or protein if you see 3 letters followed by a number. That's pretty much how we label our genes when they're discovered. And then we follow it by calling the protein the same name. And if they're going to be in the same pathway or do the same job, they will often have the same first 3 letters. So all of these proteins will either be SNF or SWI. SNF5 functions as a histone binding protein, BP, histone binding protein. So this is going to help attach it to histones. And then again, here we have our ATPase. Interestingly, there is evidence that shows that SNF5 here, this protein, will go from this nucleus to the cytoplasm and back and forth under certain growth conditions. In times of stress, this SNF5 will often leave this complex, which would prevent this whole complex from binding to histones. The next one I'm going to discuss real quick is SWI3. 2 subunits bind here, and its job is to displace H2A and H2B from the nucleosome. So when the gene needs to be activated, SWI3's job is to remove these 2 histones. Just like SNF5 here, it also will go back and forth from the cytoplasm and the nucleus. Okay, another one I want to mention here is SWI1. SWI1. And it's involved in some very specific activities in yeast, like mating type switching and some other things. But the reason I want to point this one out is because in humans, it may be, there's some evidence, it may be a tumor suppressor gene, TSG, for breast cancer. So we want tumor suppressor genes to be turned on because they help prevent cancer. But if a mutation of this prevents the ability to turn that gene on, it could lead to a misexpression of a tumor suppressor gene and cause breast cancer, or cause cancer. But in this case, the homolog in humans is thought to be linked to breast cancer. Okay, and I think this will be the last one I mention, and it's called ARP7. And I know that they will all be called SNF or SY1, but every now and one sneaks in here. And this one is important because it has a slightly different function than what we've talked about. Our proteins have a actin binding domain, so they can bind actin, and this may play a role in helping to position or move this complex as the need for gene expression comes up. Now, as I said, there are at least 12 subunits that are found in this, and these are just the ones I'm mentioning right now. Okay, I know I already mentioned this, but I want to stress it again, that these are not static structures. They change quite a bit. The proteins have to bind here in specific sequence. Sometimes they're bound, sometimes they're not, and that can lead to various degrees of gene expression. And one way that they regulate this is by having the subunits move from the cytoplasm to the nucleus. In yeast, it's pretty easy to study because you can just expose the yeast to certain kinds of growth conditions, and you can monitor the movement of the proteins between the cytoplasm and the nucleus. Another thing I want to point out is the observation that these proteins in this complex in general is in low abundance. I'm going to write it down here. Low abundance. And what this suggests is that it's not a constitutive part of the chromatin. For instance, the histone proteins are in high abundance because they are a constitutive part of the chromatin. These chromatin remodeling factors are expressed at a very low level because they're not always needed. They're not constitutively expressed. Okay, I want to spend just a little bit of time talking about tissue specificity. We're going to have to leave our good friend the yeast for a little bit because yeast, being a single-celled eukaryote, doesn't have multiple tissues, and so its level of expression is really environmentally dependent. But in multicellular organisms like Drosophila and humans, tissue specificity is a concern. It's something that needs to be addressed. For instance, if we'll pick two tissue types, let's say you have liver tissue, and then let's say you have the other sample is brain tissue. There's a certain set of genes that are turned on in the liver that aren't turned on in the brain, and vice versa. These chromatin remodeling factors play a critical role in deciding which genes get turned on or off. So it's not surprising that subunits on those large chromatin remodeling factors, that some of those subunits play a role in tissue specificity. In humans, remember, the chromatin remodeling factor that is similar to the SY/SNF complex in yeast is called BAF. And I'm just going to write that here, okay, BAF. So a lot of the proteins found in the human version of the SY/SNF complex are called BAF. And remember, and I'm going to just kind of draw a generic version of what this complex might look like. You're going to have your ATPase here, and then you're going to have other proteins associating with it. Maybe for a liver gene, I'm just going to write this as BAF-L. Maybe this BAF-L is found in liver tissue. It associates with this complex and helps turn on liver-specific genes. In brain tissue, BAF-L would not be expressed because you don't want to turn on liver genes in the brain. So in the brain, there's no BAF-L. So let me do this. No BAF-L. So it's not going to bind to this complex and turn on liver genes. However, maybe in the brain tissue, we have a protein in this complex called BAF-B for brain. And maybe in brain tissues, the BAF-L is removed or is not there, but the BAF is expressed and it goes to this complex. This complex now, because of this new BAF-B gene here, BAF-B, it now is directed to brain-specific genes. Being directed to those brain-specific genes, it'll just turn on those genes in the brain. BAF-B, not expressed in the liver, because you don't want to turn on brain genes in your liver. You want to turn on liver genes in your liver. So this is just a strategy to customize the chromatin remodeling factor in humans and other multicellular organisms. They can interchange these subunits to give it a slightly different function. It still has the general function of chromatin remodeling, but by adding a subunit here, it adds that degree of tissue specificity. Okay, let's return to the SWI/SNF complex. Okay, I redrew the SWI/SNF complex here just to remind us that it is a very large complex. And I want to just briefly mention some of the roles that it does in yeast. And at the molecular level, what it does is it binds to nucleosomes. Remember, nucleosomes were always kind of perplexed how something can bind to the DNA in order to remove it, because that DNA should be inaccessible. But these chromatin remodeling factors, the SWI/SNF, is able to do that. Nucleosome DNA, I should say. It can displace some histones like H2A and H2B. It can move histones down the chromatin. So it doesn't have to displace it completely, it can move them down. This complex in yeast is important in protecting the cells, the yeast cells, from stress. A big one that they study is hypoxic, low oxygen environments, but other stresses as well. All of these steps here requires energy. Many of these steps along the pathway here are energetically unfavorable, but there is a suggestion that this can handle the energy demands, and that is because this central unit here is a very well conserved ATPase, as we've talked about. So central to this whole complex is sort of like this engine that provides the energy to do all of these things. Now, as I said, one of the important roles for the SWI/SNF complex is to bind to nucleosomes, the DNA on the nucleosome, and make it accessible for other proteins to come in there. So if I were to draw our fabulous double helix here, and draw our histones located on here, how does this complex, the SWI/SNF, get in here? And not only that, how does it make the DNA accessible to other enzymes? Well, what I want to do is focus in on just one of these. And remember, even though it's hard to tell in this drawing, the DNA wraps around these histone proteins. The DNA doesn't go through them. So we could draw it slightly differently. And so let's do that. Let's draw our histones here. And remember, there's 8 of them. And let's draw the DNA wrapping around the nucleosomes, or the histone protein. So again, these are our histones, and this is our DNA. And as of right now, various transcription factors cannot get in there. They can't find the genes because it's so tightly packed. So what our SWI/SNF does, it comes along here and binds the nucleosome like so. And this will be hard to draw, but hopefully you'll get what I'm trying to show here. So this SWI/SNF, what it does is, as it binds the DNA here, it begins to pull it together. So the SWI/SNF kind of moves down. And as it moves down, it's pulling some of the DNA with it. It's not going to completely unravel it like a ball of yarn. It's just going to pull a little bit of it down. And so what you might have after it's initiated this is something like this. Where you form this loop of DNA that's been pulled away from the histone proteins. So it's still down here, it's still holding on to it. At some point though, it will dissociate. Now, how the SWI/SNF protein complex changes shape in such a way to pull the DNA in this direction is not clear. But we know that ATP hydrolysis is important. For one reason, it contains that very well conserved ATPase in the structure, but we also know that if we measure ATP hydrolysis during this reaction, that as ATP increases in its hydrolysis, more of the DNA loops out. So there does appear to be a strong connection with the hydrolysis of DNA. And as I said, at some point, this dissociates. And now what we've done is we've exposed this DNA to other transcription factors that could now come in and begin to pull it apart maybe even more. This is one of the beginning steps at making this DNA more accessible. But you could see how you wouldn't want to make all the DNA this accessible, because you don't want all the genes turned on all the time, even in the relatively simple organism of yeast. Okay, now let's look at this structure we drew at the beginning. One thing that might have popped in your mind is a problem of nucleosomes colliding. So let's just focus right here on this one. As we're pulling the DNA away from the nucleosomes, stretching out like so, and it's going to stretch more and more and more, right? This DNA doesn't just magically appear as it's being looped out. Where does it come from? Well, it comes from the linker DNA. And so if it was just this one pulling the DNA out and shrinking the linker DNA in between them, what would happen is that the nucleosomes would collide, more than likely. But what helps prevent that from happening is that the neighboring nucleosomes are also doing it. So it's also linking the DNA out. And I think this is hard to visualize, so work with me here for a moment. As each of these are pulling DNA out from their neighboring linker DNA, what's happening is that these nucleosomes are shifting down. It's like pulling one string, and now you're pulling this one along. Pulling one string, pulling this one along. So you can imagine a whole string of nucleosomes just moving down. One of the advantages to doing this, as opposed to just disassembling the whole nucleosome, I would assume, is to allow the histones to still be closely associated with the DNA, so that when you want to turn these genes off again, it's easier to push them back to the right place, as opposed to having the nucleosomes being kicked off here and just kind of like free floating here, waiting to be reincorporated. This kind of keeps them in the same vicinity. Another important thing about this movement is that it shifts the nucleosomes in unison. And so as it's shifting the nucleosomes in unison, you may have a large gene here that you need to expose. And by doing so, you're moving a whole chunk of this highly wrapped up DNA down the road, so to speak. Now I want to take a step back again. Seems like we're always going backwards. Let's draw our lovely model here again with our nucleosome structures, like so. We just explained how SWI/SNF can move these nucleosomes away from the DNA and how it pulls that DNA away from it. But a central question still is, how is it able to bind specific pieces of DNA? Why is it only some areas where it binds? And even in those areas where it's binding, how can it bind to these areas that are supposed to be protected against enzymes? So we know what it does once it gets there, at least we have a model of it, but how does it do that? And so I want to spend a little bit of time talking about how it does that. And in this discussion on how it does that, we want to talk about DNA sequence. We want to talk about H1, the lost histone, so to speak. We want to talk about histone variants and histone modification. So how do these factors help initiate transcription here, and ultimately elongation too? Because the goal isn't just to start this. We want to move all these histones away so that the whole gene can be transcribed. Okay, so let's go ahead and start looking at how these factors influence SWI/SNF's ability to make that first and initial interaction with the nucleosome. So let's take these first two in the same discussion here. There is some evidence that would suggest that in between these two nucleosomes, there is regulatory sequences. That either the SWI/SNF complex or some other complex can come in here and begin to open up the DNA and either move or displace these histones. But we wouldn't want these regulatory sequences to be exposed all the time, because that would take away from the ability to regulate this process. And that's where H1 comes into play. We know that H1 plays a role in compacting the whole chromatin into that 30 nanometer fiber, but also in linking these two nucleosomes. We know when H1 isn't present that we don't have the complete packing of the chromatin. We also know from other studies with mice that if you disrupt at least 3 of their H1 genes, then it's lethal. So we know it's important. But what this mechanism is suggesting is why is it important? The model would say that H1 stays here, hiding these sequences, these regulatory sequences, so that the SWI/SNF can't get in there. Because it would recognize this sequence. There's some sequence specificity that maybe this can recognize. During the times when you want to remove these histones, the H1 is removed. When it's removed, we have restored the ability of SWI/SNF to come in here and begin to unravel the DNA from these histones. But it requires this first open piece of DNA to get started. So we can think of this histone, this H1, almost serving as a gate. It either comes down here and closes to prevent expression or the removal of the nucleosome, or it opens, allowing access to this region here. Next, let's talk about histone variants. If you remember early on, we talked about this octamer here. We said there were 2 H2As, 2 H2Bs, 2 H3s, and 2 H4s. And these are the classic forms. But we know there are some variants. And we'll talk about some of these throughout the semester, but what I want to talk about now is a variant of the H2A. And we call this one H2AZ. H2AZ. And to talk about this, I need to open up a little space here. And I want to draw our DNA just slightly different. I'm not going to draw it as a helix right now. I'm going to draw it kind of like so, where the DNA is looping around, and in here we have our histone. And this middle region here that I left blank is a region we call the nucleosome exclusion element. This area does not bind nucleosomes. These nucleosome exclusion elements are usually near areas of highly transcribed genes, particularly the promoters. So that in itself, alone, without talking about histone variants yet, helps expose this DNA so that these chromatin remodeling factors can come in here and activate transcription. What is usually found flanking these nucleosome exclusion elements are variant histones. So in the histones here, they have all of these histone proteins except H2A. Instead of H2A, they have H2AZ on both sides. And then you just go one nucleosome over and you're back to good old H2A. So it's only in the regions next to this nucleosome exclusion element. The reason we have this variant is not completely understood. It is believed that it has a looser association with the DNA in these regions here, making modifying the chromatin in this area more straightforward, or requiring less energy. And this last one here are histone modifications. And I'm not going to spend a lot of time on this now because we're going to spend a lot of time throughout the semester talking about this one, but I want to introduce a couple terms. We know things like acetylation. Adding acetyl groups to the positively charged tails of these histones will loosen the interaction between the histones and the DNA. So this is a very important modification that helps begin the process of unraveling the DNA from those histone proteins. Methylation also plays a role. We'll talk in the next podcast about how methylation of the promoters decreases expression. Methylation can also occur on the histones to affect gene activity as well. Okay, I'd like to close with a summary. We began by talking about the nucleosome problem, so to speak. How do we get in there to expand the DNA so that we can have access to the genes that need to be transcribed? We talked about the role that SWI/SNF has in this process, as well as other members of its family in humans and Drosophila, and how this plays a role in looping out DNA from the nucleosome, and how that allowed for larger removal of those histones, or the sliding of those histones, so that larger areas of the DNA were accessible. And then we took a step back, trying to address how it is that this Y-SNF can have that first contact to begin to pull apart that DNA. And we talked about several things that could account for this. We talked about the role of H1, DNA sequence, particularly the nucleosome exclusion element. We talked about histone variants and how there are variations to the classic histones of H2A, H2B, H3, and H4, and how these variants sometimes have different affinities for DNA, which could help expand that DNA for transcription. And then we talked about histone and DNA modifications, just at a very basic level, and how that can expose the DNA. All right, that's all I have for right now. If you have any questions at all, please make sure you come see me. If not, I'll see you in class. Bye.